I'm here at the ASAM meeting with Dr. Ken Blum. Dr. Blum has done a study, a statistical analysis of data looking at compliance to medication and abstinence from drugs of abuse, and he's tried to correlate this to a genetic addiction risk score. Now, Dr. Blum, would you describe the study in more detail for us and tell us what exactly is a genetic addiction risk score?
Well, the genetic addiction risk score is something we call GARS, and it's something that we're developing with Dominion Diagnostics to commercialize a genetic test that goes across multiple genes that are involved in the reward circuitry of the brain. So everything from serotonin transporter genes down to dopaminergic receptors like the D1, D2, D3, D4. We look at dopamine transporter, we look at other genes that are involved in the full mechanism of how one actually releases dopamine at the nucleus accumbens. So what we look at is the risk alleles. And these are candidate gene approach. And it turns out that the risk alleles, there are thousands of papers now on each one of these risk alleles that we select. So we're looking at 10 genes, and we're actually looking at about 18 polymorphisms, what we call SNPs.
Right.
Okay, and what we do is we're trying to correlate the stratification of risk. Now, we have low risk. It doesn't mean you don't have risk, because you still got a polymorphism. So it's still risk, but we call it low, moderate, and high risk. And one of the things that are actually very interesting is that we're trying to correlate this risk with the ASI, the Addiction Severity Index.
Okay.
And the good news is that we are actually assessing the data now, and what we find is that there seems to be a linear trend of significance where the genetic risk score actually correlates with the ASI composite for alcohol abuse. And now we're looking at drugs, and we're looking at some of the other things. So we're getting a good handle as to this particular genetic test, which is really the only one that has done all the research to validate it, actually seems to be potentially important for clinical benefits, not only in terms of risk, but in terms of medical monitoring and looking at certain genes and how that could affect whether you get binding with Vivitrol. So it's also a medical monitoring benefits that could come out of that.
So you're now still doing an analysis, or you're having an analysis ongoing right now, looking at medication compliance?
Yeah, we're looking at medication compliance. There's been a number of studies already showed that certain genes can affect outcome based on whether or not methadone is working, or naltrexone is working, or whatever, buprenorphine is working. So that one of the things that the benefits of the test is to see whether or not you have these gene polymorphisms, so it may actually affect how you actually going to treat the patient in terms of dosage. That's part of it. That's just part of it. Yeah.
I see. Okay.
Yeah.
Now, for the alcohol issue, have you actually determined the sensitivity and specificity for this gene test?
Yeah. Well, of course, there's been a lot of studies out there that had ROC curves and specificity and all that study. A lot of them have already looked at genes individually. Okay, but this is probably one of the first studies to put them all together, so that in a sense you're mirroring how the brain reward system works. So we're going to be doing all of that. We're actually going to look also at every individual gene and see where that correlates.
See what the contribution is
with either the ASAM. You got a new ASAM dimension. We may be looking at that. We're going to look at ASI. We're going to try to look at these neurophysiological tests, paper pencil basically, and see where the genes fit with those tests.
Okay, very good. Thank you very much.